All right. Hi, everybody. This is the walkthrough video for Tuesday. It is January 28th and try to week 10. And what is the name of the type of respiration when cells do not use oxygen to create ATP? Remember, we learned about it last time. What is it if they don't use oxygen? If you don't remember, look back at yesterday's assignment. Today, we're thinking about data on aerobic and anaerobic respiration because we're going to put our learning to use. So today, I'm passing out the assignments for Wednesday, Thursday, and Friday. So people are going to get a lot of papers. I will make sure that the table of contents is up to date. I'm going to leave it as turquoise for this time until I'm back from class in week 11. I'm back from my trip. So you're going to get a bunch of papers today. They're going to go on the table of contents page. But today, our assignment is called bacterial broth. One thing I like to share with people are the Nobel Prizes in medicine. So the Nobel Prizes come out in October. And this year, they actually gave the Nobel Prize, this big fancy prize in medicine, for thinking about messenger RNAs, tiny little RNAs that help turn messenger RNAs on and off. So that was pretty cool. All right, here's our table of contents page. The one you need today is bacterial broth. You are also getting a whole bunch of other stuff, but I'm going to show you what that is as we go through these videos, and then it'll be live for you. So the yellow is for my class today as I'm recording this. You are recording 50, it's bacterial broth. All right, life has a lot of systems. We're thinking about ecosystems. We've learned about the carbon cycle, and we've thought about how cellular respiration and photosynthesis make that carbon cycle happen. We most recently have thought about how cellular respiration works. Even without oxygen, cells are able to do ATP, but they can't do cellular respiration. So where are we going? We're going to look at this system where you can grow bacteria in a section that has a liquid with no oxygen, and then a liquid that has some oxygen in it, but there's glucose the whole way through. And so here is a tube with nothing growing. Here are 3 tubes that have different types of microbes growing in them. And you're going to use that to be able to work on the assignment bacterial broth, the same way that soup is like broth. Microbiologists use this type of broth, thioglycolate broth, to determine the metabolic pathways used by different species of bacteria. So again, the entire broth contains glucose, a food source for the bacteria. The top portion of the test tube contains oxygen from the environment, and the highest levels of oxygen are found here. And then sodium thioglycate creates an anaerobic environment by eating all of the oxygen in the lower portion of the test tube. So it's a chemical that reacts with the oxygen and takes it away. Bacteria are introduced throughout the broth, and they grow in different areas of the test tube according to the needs that they have. So A is Anthrobacter, B is Clostridium, and C is a Staphylococcus bacterium. So a control tube has no broth but no bacteria. All 4 test tubes were incubated in a warm temperature for 24 hours. And then the medium, the bacterial growth is lighter. It's that yellowish color in this graphic here. So what you're going to do is fill this out. What is it like in the aerobic part and the anaerobic part? And again, aerobic is with oxygen. And then where do the bacteria live, if anywhere? After that, you're going to think about whether an organism is doing aerobic respiration, anaerobic respiration, or maybe can do both. Remember, this top part has oxygen, this bottom part does not. We need oxygen, but not all bacteria do. Some can do both aerobic and anaerobic respiration. So you're making a claim for each of those types. Is it aerobic respiration or is it anaerobic respiration as to how those are utilizing matter and energy? Then you're going to describe what you see in the picture that helps you make that explanation. And the reasoning part is like the common sense part. Well, you only see growth, maybe you have a blank tube and you're like, yeah, those bacteria didn't grow at all, right? That's not what happened, by the way. You're not analyzing this control tube. You're only analyzing these. I'm trying not to give you the answers. So after that, imagine that scientists have collected a species of bacteria that live with these tube worms in these deep sea vents. These bacteria are the major producers. They are the things that get eaten, that then get eaten, that then get eaten. And scientists want to figure out, is it photosynthesis or is it chemosynthesis? So if you were absent, try to think about how you would use this type of test tube plus or minus a light to figure out if the bacteria are photosynthetic or if they're doing something else. In this case, doing chemosynthesis, getting their energy by manipulating the materials near the sulfur vents. So that's what's happening at the end of this assignment. I'd like you to try your best on this one if you weren't here. All right. Last but not least, I wanted to share with you the bell work. We are indeed skipping the bell work because today in class, people got a lot of papers. See the Canvas videos for each day. If you end up absent as well, please just use a piece of regular paper and you can take the notes, or you can print out the sheet and do it at home, or you can come and pick up a paper from our extra pages box. All right, that's it for me today, but I'll be back with a video every day this week. Take care out there. See you soon. Bye.